it going?
Good, how are you?
Good, thanks. All right, we're just gonna let a few more people join. Okay. So. All right, we got a couple more people just getting ready to come in, so let's just give it one more second. Alrighty, guys. Well, good afternoon. Welcome to week 7. As always, I just want to start out with a quick poll, see how things are going. Some of you guys that have already had me today have already been through this, so just want to get a feel for how we're doing in class and life. I'm more worried about life than this class, honestly. So
True. Just the uncertainty, is really, I think, getting to a lot of us.
Like, in what respect, Zoe?
Just kind of like, I feel like everything's in kind of a limbo right now, especially with school, because, have they officially confirmed that next semester is all online too?
I don't know what you guys have been told, but so here's the sitch. And I don't know what part of this is privileged information, and I also don't care. So I just am over kind of everything at the university level management right now. So we know that spring will be mostly online. And my colleagues, especially on the ANSI side, I can't speak to other disciplines, are only gonna really push for in-person experiences for individual research projects or internships or like apprenticeships out at the farm at the animal units, those type of things, because we saw what happened when we pushed for in-person labs and they kicked us out, and it was hard on us, but we were distraught over how it was affecting you guys. And so we've just decided that knowledge is power at this point in time, and so anything that we can control, and as far as what's going to be online and what's not going to be online, that we're going to do this, because it's the uncertainty, you're absolutely right, is just ridiculous. So I would say that unless you are interested in doing an internship or what is going to be the old ANSI 110 model, or an individual research project with an individual faculty member, that you're probably going to be online next spring. So I'm not sure how many other classes are going to fight for in-person stuff, just because it got taken away like really, really quickly. And so we just lost a bunch of time in planning, and we saw the stress it had on you guys, especially those of you guys that like moved to Chico and signed leases and then had to, and I really feel bad for like the freshmen in the dorms. So we're just I don't know. I'm very much in the position of whatever we can control, we're going to control right now. And so I personally can control my classes in the spring, and I've chose to push all those online. So I'm just it's not the best experience. It's definitely not what I want to do, but again, knowledge is power at this point from my perspective. So that is not an official university announcement. I will deny it. Okay. So that is the University of Casey right now. Okay. So just be aware of that. So all right, cool. Well, I'm gonna go ahead and end polling here. And we're bimodal today. Bimodal. So part of us are still in ugh, this is hard, absolutely. Some of us are getting time management down, and some of us have found a groove. So that's cool. I think for me, the only blessing in all of this is I finally have a routine. And that's the blessing and the only blessing. So I get you guys. So hang in there. Again, if you guys are struggling with classes and you're getting overwhelmed, keep in mind that the university is going to allow you to do or is going to allow you to drop classes clear up till the last week of the semester if needed. And so I have been advising several people that normally take 15 to 18 units and are just ran ragged right now and not doing well in any of their classes to actually, on a case by case basis, drop a class just so it's manageable. So I think right now, like, let's, just salvage. It's straight triage right now, right? And so we're definitely in triage mode. So all right. Well, thank you guys for answering the poll. We'll get that out of here. So I have a couple of things that I want to go through, and then Zoe has a question for us that I have prepared for Zoe. Like, look at this. And I even brought in props. Actually, I just stole them out of the yard. So we're going to come back, and Zoe can ask her question about pseudo replication after we get through announcements and stuff, okay? All right. And thank you guys for using your cameras. It's so great to see your beautiful smiling faces. All right. So you guys saw in the email that came out on Friday? Is that when that happened? Let's go with Friday. That we've got kind of we're headed into kind of a heavy area, right? So we are headed into statistics, also known as sadistics, for those of us that like to call it what it really is. So I'm just gonna go ahead and share my screen with you, and we'll get onto Blackboard and figure out where everything is at and what we got going this week. And I have my tablet going over here, so now I don't have to ask like what you guys can see and what you can't. So for the moment. Okay, so in our start here folder, we are in week 7. So crazy, so crazy. And so we've got quite a few things happening this week from a lecture standpoint. And we've got 2 big lectures that we're working through. We're working through an introduction to statistics, which has 5 parts to it. So 5 video lectures. And they're between like 7 and 12 minutes, I think. And I tried to split them up. So we were talking about specific points. So if you need to go back and listen to one certain thing, you can. And so this is really just, I'm going to open the note guide for us so I can kind of tell you what we're talking about. But this is a reintroduction of statistics. So I know that you guys, it's a requirement for our college and stuff, but I know you guys have been through Math 105 or the equivalent of that at a junior college, but let's be honest, how long has it been? A while? Probably a while. right? Okay.
Freshman year for me. Freshman year.
Freshman year. Yeah. So I finally came to the conclusion after about 2 years of teaching this class that I just need to reteach statistics. And so that's what this lecture is. All right. And so you guys have partial notes in the notes guide, but it is going back through what the different types of statistics are. And I went clear back to the different types of studies that we have. So this is stuff, observational and experimental studies that we talked about in week 1. We're just revisiting it and bringing it back up today because it directly relates to the types of data that we're going to collect out of these, different research projects. And so we talk about the types of data, examples of the different types of data, and then we start talking about how we describe populations. And so keep in mind that a population is going to be, every alfalfa field or every beef cow in the world, right? So it's not necessarily feasible or doable and realistic to actually do a research project on every single beef animal in the entire world. So to do research on that, we actually will take a subset or a sample out of a bigger population. And if that is sounding familiar, that's good. It should. Okay. So we've talked about this a couple of times before. And so you probably remember this diagram actually, where we talked about the bigger population, and then we're going to pull samples out of that population to serve as experimental units in our research project. And so this descriptive statistics lecture is going to take you through back to the basics. So what is a mean? What is a median? What is a mode? Okay, what is variance? What is standard error? What is standard deviation? And then we get into things that might be a little bit newer to you, and it's actually leading us into this concept of hypothesis testing. So determining if the performance in one treatment group was actually different than the performance in a different treatment group. And so we start to investigate that using a 95% confidence interval. And so that confidence interval there's several slides explaining it, but it's really going to end up being a bunch of information that you're pulling together from multiple sources. Okay. So just if you guys haven't watched through the videos yet, I'm not expecting you to. It's Monday. Stuff isn't due till Friday, right? I'm going to encourage you to start early and start often and spend some time with these video lectures and starting to pull things together. And I will give you a hint. The quiz is straight up stats, okay? So have a calculator, have scratch paper, have your notes in front of you, okay? So have everything there that you could possibly need, okay? And so that is a look at the introduction to statistics. And then I think next week, guys, is on the bubble for me because I'm in the process of moving and I'm not 100% sure I'm going to have good internet next week. So I will let you know if we're having this afternoon session next Monday or not. Okay. And so I'll let you know that on Friday. I just can't 100% say for sure right now. All right. Make sure I know where we're at. So if we go back to our week 7 guide, these first 5 videos all have to do with that intro to statistics lecture. And then the second lecture is going to be statistical power. And this one is short. And so it's only 2 videos long, but statistical power is a mechanism for us to figure out how many experimental units we need per treatment. And so I go through and I talk to you about what power is, how we calculate statistical power, and then there's an example in there as well. And so that is what this week's lecture material is over. And so it might seem like a really, really steep climb uphill, especially if you have apprehension to math and mathematical functions. But I've got to tell you guys, I found a home with statistics. I was totally that kid that was kind of shamed during grade school because I wasn't great at math. And then as soon as you shame people, that makes them like shut down even more, right? So I had this big fear of math. I hated it until I found statistics and research. And so I'm hoping that this gives you a little bit more confidence as you move forward. And I'll tell you, you guys already know more than you think you do. You're going to get into that intro to stats lecture and you're going to be like, oh, okay, I got this. I remember this. It's coming back. All right. Okay, and then we've got a quiz here. So just keep in mind, quiz is due this Friday by 5 p.m. So that is what is due this week. All right, so is everybody good with week 7 stuff? Any quick questions? Dan's got a thumbs up, thumbs up. Okay, cool, cool, cool. All right, so let me back out of here really quickly, and I'm going to go back to the research project folder. And I did some updates today. And so the first thing I updated was this big, huge assignment. Okay, so it's going to eventually show up here. There we go. All right, so you guys probably remember this, right? So part 1, this was part 1. You guys already turned it in. I'm in the middle of grading it right now. So I'm working on it. It takes me some time. I'm sorry. And so I don't want you guys to stop though. We're going to keep pushing forward. And so one thing that I did change here is that part 2, which is includes the title, author, institution, introduction, materials and methods, and lit cited, was originally due basically in a little less than 2 weeks on the 16th. Changed that this morning and pushed it a week to the 23rd. All right, so the next paper assignment, part 2 for the 490 class is due Friday, October 23rd by 5 p.m. Okay, and it'll be just like the submission process will be just like what you did for part 1. There will be a submission link up. It's going to have a rubric attached to it, and you guys are going to just upload your PDF version, okay? So I just want to make sure that we're all on the same page. So I'm just going to really briefly read through what part 2 entails and give you the opportunity to ask some questions just in general about the assignment. We're going to talk about the specifics of each individual project in a little bit more detail in a bit. All right, so part 2 is going to include all of those major parts. This is really the first chunk of that paper. Okay, so some things to keep in mind is that as you develop these titles, and most of you already have because you included it as I suggested in the outline, okay, so you've already been through this process. But if you didn't, the title needs to include a minimum of 3 descriptive terms. And you guys already know what those are: independent variable, dependent variable, experimental unit. Okay, so your title can read very, very similarly to the objective of the study. Okay, it's all about consistency. The introduction section needs to, or it already has the framework based on the outline that you touched base or that you already turned in, right? So you've already got it kind of sketched out. So it needs to make sure that you state the problem, have a hook sentence. It contains the literature review, which were your themes, right? And in that last paragraph, it needs to state the objective of the research project. Okay, so the part that you haven't had any experience with yet is the materials and methods. And so materials and methods are going to be a big ask for this part 2. And so that section of the paper needs to describe the experiment in enough detail that another person can actually replicate the study. So the materials and methods portion of a paper has to have quite a bit of detail so that somebody else can actually replicate the study. So it should include a description of the experimental units, the independent variables and the different levels of those variables, the dependent variables and the procedures used, as well as how the experimental design and the statistical analysis. Okay, and so a lot of that seems like really new information, and we're going to build on it in lecture. You guys are going to get more about this stuff next week as well. But if we go back to the main research folder, keep in mind that you have these project folders with descriptions in them. So hopefully it'll download. Come on, buddy. All right, so you guys probably used a bunch of this background information for the study, okay? And then this is where we start the materials and methods. And so notice that it is in bulleted form, and there's a lot of really specific information here. Your job, and I'm going to help you with this, is to get all of this information so that it reads in paragraphs. Okay, so first and foremost, lists do not occur in materials and methods. Okay, so there's no lists in materials and methods. And so we're going to work on this over the next couple of weeks. And we're gonna outline the materials and methods just like we did with the introduction section. And then you guys are going to get the opportunity to kind of start pulling some of this information together. All right, so that is part 2. And then keep in mind that we're just going to keep a running list of literature cited sections as well. So,
Dr. Diatley?
Yes.
For part 1, we did like a you hosted like a writing hour where we could ask you questions. Can we please do that again? It was really helpful.
Yep, I will. I have them worked in. I think I can do maybe even 3 of them the week that it's due.
Okay, those are really helpful.
Yeah. They are. And I like them because you guys start asking specific questions, and I can tell those of you guys that are like actually working on it. So yes, I will make sure that writing workshops are on the list. I'm going to write it down right now.
Thank you.
Yeah. All right, cool. So 2 big things happening. All right, so quick questions about what is expected this week or the part 2 of the research paper. All right, cool. I'm seeing shaking heads, so we will roll with it. Okay, Zoe, bring on your question.
Okay, so with the pseudo replication, I thought I understood it. And I I understand the plant example, which it looks like you were gonna kind of give a visual of. And I understand that because even if it's different leaves on the same plant, it's still the same plant. So, but I was hoping you could give like another example. And then also I wanted to talk about the quiz question.
Okay. How about we do, let me, just so everybody's on the same page, let me go through and just talk about what pseudo replication is, even though you think you have a good handle on it. So everybody's on the same page. And I killed some plants, so let's use them. Okay.
Might as well.
All right. And then I think I can do what I've got the quiz question actually drawn out. So I'll bring up the tablet and kind of draw it out. And so hopefully that will help. Okay. Perfect. And then if you still have questions, we can definitely handle that. So, okay. All righty. So now the fun part. All right, so you guys can see 3 plants, right? Okay. Yes. All right, so this concept of pseudo replication is related to that principle of experimental design replication, right? And so replication is really, really central to making sure that we have enough power in our study that we have the ability to detect a difference if there's a difference there to detect. Okay, so those principles of experimental design are kind of fast held standards when it comes to designing a research project. So replication has to do with how many experimental units you actually have. Okay, and that becomes really important because we want to make sure that our individual experimental units are independent of one another. All right, so for example, let's say I have my 3 plants here, okay? And let's say that our treatment is watering regime. So this guy gets watered once a week. This guy gets watered, actually, hang on. This guy gets watered once a day. This guy gets watered once a week. And this guy gets watered once a month. And side note, if you live in Casey's house, this is you, FYI, okay? All right, so we have, and this is our whole study. There's no other plants involved in the study right now. Okay, so those are our independent variables or our treatment groups. Our dependent variables are going to be the traits that we're going to measure off of this. So just to make it kind of a relative plant example, let's say that we are going to measure surface area of the plant. So leaf surface area. So if I take this little plant here, we can pull it off, run it through a computer system, and the computer system will tell us exactly how much area is associated with this leaf, okay? So there's fancy stuff that can do that. So first of all, my plant people in the room, why would that be of interest? Okay, so we will always want to relate stuff to application in the field. So why would surface area of the leaf be important to measure?
Greater
The amount of light.
photosynthesis.
Okay, greater amount of light. I think I heard it twice, right? So the more plant or leaf material you have, the greater potential for photosynthesis you have, right? So the faster it can grow, all of that good stuff. All right, so that is why leaf area would be important here.
Could I add something, Dr. Diallo?
Sure.
So the greater surface area there is, the more stomata there's going to be on the underside of the leaf. So the more that you have transpiration, stuff like that.
Yep, exactly. So we like big leaves, but they don't write songs like that, right? So I know, and for those of you guys that know that song, it's highly inappropriate and super old. So that's why I get it and you may not. All right, so we are going to measure, let's see, leaf area on 3 leaves on each plant, okay? So as we do that, let's just jerk them off, okay? We're gonna measure that guy, we're gonna measure that guy, and we're gonna measure that guy, maybe. Okay. And then measure you, you, and you. And over here, we'll measure you, you, and you. Okay. Gosh, this is hard. This is the struggle in online teaching right here, guys. Okay, so not that you can see those leaves, but now you can. Okay, cool. All right, so this guy, keep in mind, he got watered once a day. This guy got watered once a week. This guy got watered once a month because he lives at Casey's. All right, so as at the end of our trial, let's just say that we did like a 2-month trial, right? We pulled off these leaves and we're gonna put them in that computer system and it's gonna tell us how much leaf area we have. Okay, so this is a pretty straightforward, simple research project, right? But the problem here is that we have committed pseudo replication. And pseudo replication is where our individual experimental units, which we're treating as these leaves, are not independent of one another. So the reason that we have pseudo replication in this example is because this whole plant got watered once a day. This whole plant got watered once a week, and this whole plant got watered once a month. Okay, so these individual experimental units actually were taking on water from the watering regime associated with each plant. Okay, so they weren't independent of one another. And so that means that this entire plant becomes the experimental unit, not the leaf. And then we have one experimental unit for treatment 1, one experimental unit for treatment 2, and one for treatment 3. So we don't even have enough power to tell if there is a difference between the treatment watering regimes compared or not, because we only have an N of 1 rather than what we thought we had, an N of 9 or an N of 3 per treatment. Does that make sense? So these leaves here are not independent of one another. Okay, so that's what pseudo replication looks like. Okay, so how would we change this so that we weren't committing pseudo replication? Do you guys have some suggestions?
More plants.
Oh, go
More
ahead.
plants, okay?
Yeah.
More plants, what? Be more specific.
For each different water regime.
Yep, so we would probably want to have a minimum of 3 plants for the once a week or the once a day, once a week, and once a month, right? So we'd want to stack them up like this so that all of these guys were going to get watered once a day, once a week, or once a month. Then we have individual treatment groups. Okay, so is everybody okay with what pseudo replication is?
Can you explain it one more time? Because I'm having a hard time understanding why if you add more plants, it's changing it from an experimental unit to pseudo replication.
Yep. Okay. You guys are going to pretend like fake cactuses are these plants, okay? All right, so, and I'm out of plants. Crap. Hang on. Plants don't live long at Casey's, FYI. Okay. Hopefully this will work. It's a numbers game, Rebecca.
Okay.
So this is what we just suggested, how we would change it, right? So we went from having only 3 plants to 9 plants total, 3 per treatment. So these 3 are going to get watered once a day. These 3 are going to get watered once a week. And these 3 are going to get watered once a month. Mm-hmm. So per treatment, we have 3 independent experimental units because this guy holds its own water, right? Mm-hmm. This guy holds its own water. And this guy holds its own water.
Yeah.
Okay, so another way to kind of think through this for you animal people in the room that you like see plants and automatically think black thumb, I get it. I am that person. Okay. View these cups or these individual plants as animals. Okay, so in treatment 1, we'd be feeding 3 sheep. In treatment 2, we'd feed 3 sheep. In treatment 3, we would feed 3 sheep. So we've just increased the total number of experimental units, and we've made it so that we apply the treatment directly to the experimental unit. Okay, so that when we go and if we go back to the plant example and we go and we measure surface area of the leaves, we take 1 leaf from each individual plant. And then it's truly, truly independent.
That makes sense. I'm just very visual. I couldn't visualize it. I'm sorry.
Nope, that's okay. I'm hoping this worked because I just killed a lot of plants. So
I understand the animal model more than plants for some reason. I don't know why.
yep, I get it. I get it. So all
Thank
right,
you,
cool. So you guys good with what pseudo-replication is?
So would the experimental unit be the treatment or the individual plant?
So experimental unit is the individual plant.
Okay.
So the experimental unit is always the smallest unit that the treatment can be applied to.
Okay.
So we can water this guy once a day. We can water this guy once a day. We can water this guy once a day. Okay, but back when we had the original problem, ignore them, and we had the 3, we could only water this 1 plant once a day. We had to water this 1 plant once a week and this guy once a month. And so we were trying to collect the same amount of data points, but we were treating the leaves as experimental units, but they weren't truly independent because they were all attached to the same plant. So they all had the potential to suck up the water. Does that make sense?
Yeah.
Okay, cool.
Well,
awesome.
That kind of leads into the quiz question then, because I'm pretty sure the quiz question gives the answer that like the entire group of mink was the experimental unit, not the individual ones.
Yes and no. So let me attempt to draw it out, okay?
Okay.
All right, so let me first show you where we're at. Okay, so the question that we're going to go through, that we're going to work through, and Zoe, I'm assuming this is the one that you talked about, was about the mink, right? So the question reads, Myrtle's marvelous mink is the top mink coat producer in the Western Hemisphere. Grandma Myrtle recently noticed an increase in the cannibalistic behavior causes damages to the pelts, reducing the value of the Myrtle's mink coat products, and it kills the animal. Grandma thinks that the behavior is caused by a cysteine deficiency. Grandma's aspiring young scientist grandson, Fred, believes it's a methionine deficiency. The two decided to collaborate on a project to answer this deficiency question. They decided to use 24 pins of mink kits of similar size, so 4 kits per pin, 96 kits total, in 3 categorized physical builds or sizes. So small, smaller, smallest. Okay, the Myrtle team decides to compare a negative control, a methionine treatment, and a cysteine treatment, examining pelt size, color, coverage, and density. So if we're wanting to avoid pseudo-replication, how should Grandma and Fred define their experimental units? All right, so let me attempt to use the tablet. You have to do this. Okay. Whiteboard share. This is trippy. Okay. So in this example, you guys are going to have to talk to me because I can't see the chat, but what is the independent variable? What is the independent variable?
The number of kids? The number of kids?
Okay, so not quite. Good guess. Keep in mind, what is the independent variable? Not necessarily in this example, but just what is it if you had to define it? All right, guys. So I'm going to be a little bit straightforward with you, okay? In that this is the stuff that we should start knowing off the top of our head, okay? Okay, let's switch gears. What is a dependent variable?
Isn't that the response? The dependent variable is the response to
Yes,
treatment?
it is. So, for example, in this example, what would it be?
Would it be their deficiency, whether it's cysteine or the methionine? Sorry if I'm not saying that right.
Okay.
No, would it be a cannibalistic response that's ruining the pelts and killing the other kids?
So that's the problem.
Oh, okay.
What you described first, like the methionine, cysteine, that type of thing, that's actually the independent variable. Those are going to be the treatments.
Oh, okay.
Okay. So our independent variable is going to be that negative control, methionine, and cysteine treatment groups, right? So in this example, the minks are going to get one of three treatments, either the control. Let me redo this. Okay, that doesn't work. Oh my gosh, I can't work my thing. Hold on, guys. Good night. No, eraser off. Okay, sorry. So control, methionine, and cysteine. Okay, what's the dependent variable going to be?
The dependent variable is the response to treatment. So,
yep. So there was like 4 listed. Just give me one of them.
I don't remember what the 4 were listed. I'm sorry.
Okay.
Would it be reducing the cannibalistic behavior?
So that's what we're hoping to happen. Great guess. But so Matt, actually, you're right. So it's like color, pelt size, etc. Okay. Gosh dang, I cannot make this work at all. Okay. And so the next question there is going to be, what's the experimental unit, right? And so with the experimental unit, that is where we get into this issue here. So first of all, how many total experimental units are we dealing with?
3.
Okay. So that's the independent variable. That's the treatments.
So 4 instead, because that would be the, no, I'm sorry. I'm going to stop. I don't know.
Okay. So what are the individual mink kits? Do they sound like the experimental unit?
Right? I think Are you asking for a number?
I was originally, yeah.
I think there's 96 of them individually.
Yep. So our experimental unit is going to be the mink kits, right? So these are going to be little mink things that eat food, right? So that's going to be the smallest possible unit that the treatments can be applied to. And keep in mind, our treatments are our individual or independent variables. So what we end up with here, based on what was described in the study, is that we have 24 pins Is that right? 1, 2, 3, 4, 5, 6. Yeah. With how many mink per pin? Is the problem that we can't remember what the treatment or the question actually asked?
Yeah, was
Is
it
it 8?
4?
I think 4.
4. I'm not going to do this for the whole thing, but you guys get the idea, right? Okay, so we're going to have a total of 96. Okay, so big N is going to be 96. And then a little n, which is the n per treatment, is going to be 4 per pin. All right, so the question is how do we avoid pseudo replication here? And the issue is that these little mink dudes are individual animals, right? So they have their own will to eat. They are going to have their own performance. They're going to have their own color, their own pelt size, etc. But can we, in these pins, do we have the ability to individually feed every mink?
Yeah, we could if we gave them all different feeding pins and watched them eat.
Okay, but did the problem say that?
No. So in the
Okay.
problem, we don't have that.
Yep. So we have to change our experimental unit to a pin. Okay, so this guy is an experimental unit. This guy, and this guy. Okay, so that this pin here is going to be the control. They're going to be fed the control ration. Okay, this pin here is going to be fed methionine. This pin is going to be fed cysteine. And then we start over, right? Control for all 24 pins. So that's how we avoid pseudo replication here. Because if we do this still gives us plenty of power or plenty of experimental units per treatment to actually detect a difference if there's a difference to be detected, right? So out of 24 pins, and we have 3 treatments, that means that we're going to have 8 pins per treatment. Does that make sense?
Yeah.
Okay. So this example was very much trying to avoid pseudo replication because, like Rebecca said, our initial thought process was to try to feed these guys individually, right? But we don't have the setup to do that unless we gave them their own little independent food bowl and put them in their own pen and watched them eat. We have to apply the treatment to the whole entire pin. All right, so I'm hoping that helped. Zoe, what are your thoughts? Are you more confused or less confused?
No, that makes sense. So technically, the individual mink could be its own experimental unit. But for this problem, not, because they wouldn't have like the setup for it.
Exactly. So the experimental unit here becomes pin.
For this
of 4.
I was confused because I just thought, because I thought that, like, you know, like you mentioned with the plant example, that, you know, if we can have just one individual, like, plant or animal or whatever as the experimental unit, that is the best, right? Like the smallest.
Yep. To increase power.
Yeah. So that's the answer I selected. And then when I got it wrong, I thought I was misunderstanding what the experimental unit like rule should be. That if I think I just didn't apply it to this specific problem.
Nope, that's okay. And so this was a little bit of a big ask for you guys, but I want you to start getting used to seeing these big detailed problems. Okay, and so with lots of information coming at you that you have to balance. So that's also why there was only one of them in this quiz. Okay, so just keep using these as like learning experiences, and we're going to start building on this over and over and over again, because this is the type of situation that you're going to see like on the final exam, where I just give you a paragraph of details of a study, and you've got to tell me what the independent variable is, what the dependent variable is, what the experimental unit is, and, you know, the experimental design, which is where we're headed. We're headed to experimental design. So, okay, I hope that helped. We're over-ish. So I can hang out for 6 more minutes if you guys need. Actually, I'm hanging out till 4. So, or till 5 actually. So if you guys have other questions, feel free to jump in. If not, we will see you hopefully in a week. And just remember to get that quiz done by Friday at 5. All right.
Thank you, Dr. Diatley.
You're welcome. Thank you. There we go. That's better.
Thank you.
You're welcome. Thanks.
I have one more question, but it's now about 3:40. So if anyone
okay. Bring it on.
I started like working more on the worksheet, so I wanted to come back. It's just, oh no, I have to pull it up. Okay, here. So is GnRH the only hormone receptor found on the gonadotropes?
No, there's like inhibin receptors on there as well. There's a bunch of different stuff, but GnRH is the reproductive hormone that is going to be doing most of the regulation of LH and FSH. So it's the one we talk about.
Okay, because I couldn't really find any others in the lecture except for GnRH.
Yeah, because that's the one that we're going to concentrate on. But so like inhibin comes from the follicle and feeds back to the anterior pituitary in the gonadotrope cells and will shut down FSH.
Okay.
So there's lots of receptors on these cells. Just in terms of reproduction, the one that we talk about is GnRH.
Okay, cool. That was the only other question I had, I think.
Cool. Thanks for bringing up the pseudo replication thing. Yeah,
I just wanted to make sure that I was understanding it, because I